All right, welcome back. Here's our last installment of our Chapter 7 material. This is covering a process that we call horizontal gene transfer. Sometimes it's called lateral gene transfer. If you go into other resources, you might see that phrase. But basically what the idea is there's a couple different processes that bacteria can take advantage of to share genes across the same generation. And the reason why it's called horizontal gene transfer is to differentiate it from what we call vertical gene transfer. So if you're a bacterium and you go through binary fission and you make daughter cells and those guys make daughter cells, this is what we call vertical gene transfer. That's the expected transfer of genetic information from parent to offspring. In horizontal gene transfer, you might have organisms of the same generation, so to speak, sharing genes this way. It's not a parent-offspring relationship. It's maybe like a sibling or a cousin relationship. So this is the different, we're gonna have three mechanisms we're gonna take a look at for horizontal gene transfer. Actually four, but the fourth one is kind of like a special, it's special, transposons. They're pretty weird. Okay, so let's get started. The first one is called transformation. Again, you may have seen this before. It's a topic that's covered in general bio when we're talking about history of DNA and the discovery of what actually codes for genetic information. But we'll mention it again. This is a guy named Griffith, experimented with pneumonia in mice caused by a bacteria called Streptococcus pneumonia. And he made some observations that there's a couple different strains of this particular bacterium. One strain, when he injected into the mice always died. So these were his observations. And he called this the S strain. So this is the S strain. Oops, white's not going to show up really good. So this is the S strain, and the S strain equaled death. So then what he did was he killed them. He took some of the S strain and he heated them up and he killed them, and killed S strain, he observed the mouse does not die. And then he also looked at an R strain, and the R strain didn't have a capsule. And R strain was not pathogenic. So something about the capsule made the bacteria pathogenic. And he could see this under the microscope. He could culture the bacteria, smear it out on a slide, look at the bacteria under the microscope and see that the S strain that kills the mice has a capsule, and the R strain that doesn't kill the mice doesn't have a capsule. Something special about the information that codes for the capsule, or the capsule itself, gives it pathogenicity, which we knew that. Capsules resist phagocytosis, and they're sticky and they can stick to surfaces, and so it's part of their virulence factors. So those were his original observations. Living S strain kills the mice, dead S strain, mouse lives, living R strain, mouse lives. So he's like, okay, so what if I took the two things that cause the mouse to live, right? The living R strain and the heat-killed S strain. Both of those individually, the mouse survives. He mixed those together, he injected them to the mouse, mouse dies, and he cultured the lungs from the mouse, and lo and behold, what did he find? He didn't find either of the two things that he started with, which was the living R strain and the heat-killed S strain. He found living S strain. He's like, what happened here? So he furthered his experimentation. Instead of using a mouse, he actually did it in a test tube, this in vitro transformation. So what he did was he, again, mixed some S strain with some R strain. He mixed those together. He let them sit in just a test tube. And again, he looked at that culture and he saw living S strain with the capsules. He's like, okay, so something about the DNA codes for the characteristic of the capsule. And not only that, but the living cells can take up these environmental pieces of DNA, incorporate them into themselves, and start expressing whatever genetic information was on those little bits of DNA. So that's what we come up with now, or he came up with the idea of transformation, is bacteria can take up environmental pieces of DNA, incorporate them into their chromosome or maybe on a plasmid, and express those genes. So what actually was happening was the R strains were picking up pieces and bits of the killed S strain's DNA that had the gene for the capsule, incorporating that into their chromosome, and they were expressing the capsule. So they're actually transformed R strains, original S strains, but they showed the same phenotype because they had the ability to produce the capsule. All right, so this is what we call transformation. The second mechanism of horizontal gene transfer uses a vector, a virus. In this particular case, it's a virus called a bacteriophage. And bacteriophages are viruses that only attack bacteria. They're not dangerous to human cells. Their target, their host, are bacterial cells. So in a normal bacteriophage infection, what happens is, let me see if we'll draw this on this little red virus here. So bacteriophage has inside of it viral DNA. The alien ship-looking landing structure of a bacteriophage does not go into the cell. Just the genetic information is injected into the cell. Once that does that, the cell starts turning into a viral-making factory and basically kills the cell. New viruses are made with all of their own little copies of the viral DNA inside. Kills the cell, and now all the viruses burst out and go off and infect other bacteria. So that's how a normal viral infection works. We're going to see that in chapter 13 more specifically. In transduction, a little mistake takes place. So here's step one. We have our step one and step two. We have our viral phage DNA. So the viral DNA that comes into the cell triggers breakdown of the host bacteria chromosome, and that's a normal part of viral replication. And so in the process of the viruses kind of encapsulating all of their viral DNA, accidentally, randomly, some of that dead bacterial chromosome gets incorporated into a virus. So it no longer is carrying instructions for a virus, it's carrying parts of the instructions of the dead bacteria. So then the cell bursts, the cell dies, all of the bacteriophages go off and land on a new cell. But if we're going to track this little guy, it's going to go in and land on an unsuspecting cell. It's going to think it's injecting viral DNA, but it injects a bit of the dead donor's DNA into this recipient cell. But it's not a viral infection because it's not viral DNA that's being shared. It's just a little chunk of the dead bacteria's DNA. And that can get incorporated into the chromosome. And now we have a transduced cell. We have a cell that not only has its own genetic information, but whatever that chunk is coming in from the dead bacteria is now part of its genome and will forever be part of that genome every time that cell goes through binary fission. So if this particular chunk of DNA that got picked up by that virus has certain genes on it, say for a capsule or a resistance factor or a different operon, now this transduced cell now has that genetic information. So this is what we call transduction. Okay, that's two. The third one is called conjugation. So we get to take a look at that conjugation pilus that we saw and identified back in chapter 3, that structure that's produced there. So if you are a cell that has a plasmid that contains what we call the F factor, the fertility factor. Remember we listed those at the beginning of chapter 7. So on this little plasmid, there are the instructions to grow a pilus. That's the gene, that's what it codes for. This conjugation, this fertility factor allows the cell to be able to grow the pilus. So an F positive cell, right here, has the fertility factor. It's called an F plasmid. The fertility factor allows it to grow the pilus. And then what it does once the pilus is formed is it starts to send pieces of that fertility factor over to its neighbor, right? Through these, right? So if here's my DNA and here's my pilus, it's just going to start sending that DNA through the tube. That's what a pilus is tubes. They get really close together, start sending the fertility factor over. And then as long as the pilus stays connected, the information will continue to transfer over. If it gets broken by jostling or just it's in the environment, it will break, and only as much that got sent over will be into the new cell. Now, if the fertility factor, as we're sending over this plasmid, if the gene that codes for the pilus comes over with that information, your new daughter cell can also be considered F+, because F+ means you have the fertility factor, and now it has the ability to produce a pilus when it didn't before. So now now we have a population where there's 2 bacteria that can produce pili to go and share genetic information with more than just one. Then we just started with one. So this is called bacterial conjugation, where the F factor stays separate as a plasmid. There's a slight modification to this. The next slide here, this is called conjugation with an HFR. HFR stands for high frequency of recombination. So let's take a look at what happens. So we start off with an F plus cell. It has the ability to produce a pilus, just like we saw before. But take a look at what happens. In this particular cell, the plasmid pops itself into the chromosome. So instead of staying separate as a plasmid, it is now part of the permanent chromosomal information of that HRF plus cell, and it now becomes HFR. HFR, high frequency of recombination. The reason for that is now every time that cell divides, all of its daughter cells are going to have that fertility factor, where before it was just stuck on a plasmid, some daughter cells may not have it and they would just be a regular cell. But once it's in the chromosome, it will forever be a part of the chromosome. And so now we have this HFR cell that can produce a pilus. And once it's made the pilus, it's connected to the F minus cell. Not only does it start sending over the fertility factor, but it starts sending over part of the DNA, the actual chromosome. So now not only does it have part of the fertility factor, but it also has parts of the original bacterial's DNA. And not only that, it gets incorporated into the chromosome. So this is a recombined cell. But if you'll notice, it says it's still F negative. And in this particular instance, the reason why is because the fertility factor did not get carried over. The gene, so part of the plasmid got carried over, but the actual gene that codes for the pilus didn't. So even though this is a recombined cell with a little bit of DNA from the donor, the original HFR cell, it can't make a pilus because it didn't get the gene for the pilus, but it could have. I mean, it's random. And as long as that pilus is connected, it will continue to send out as much of that copy of the DNA. So it could get a whole new bacterial genome if the pilus is stuck together long enough. All right, so that's our third type of horizontal gene transfer. So we did transformation with loose environmental pieces of DNA that can be picked up and utilized by cells. We did transduction, which was the bacteriophages inadvertently transporting bacterial DNA. And then we have conjugation with or without HFR. So it's a fertility factor allows the conjugation to take place. Now, a lot of this sounds a little bit scary, like, oh my gosh, bacteria is gonna be transferring all these genes and the bad genes and the virulence and the antibiotic resistance and all that kind of stuff. Now, this is mechanisms that can allow that to happen, but it's very rare. It's only like, I wanna say like 1 to 2% of all bacterial populations can do horizontal gene transfer, which is, a pretty small percentage, but there's a lot of bacteria out there. So we know this happens in a small percentage of the bacterial population. Okay, the next couple slides, just to wrap up on chapter 7, I wanted to talk about using conjugation as a way to map genomes and plasmids of bacteria. So these are some examples of genetic maps of E. coli. The purple one is from your textbook. That's just a regular old E. coli. The one on the black is from the internet. This is from E. coli 0157H7, which is the really bad form of E. coli poisoning, like food poisoning that can damage kidneys and stuff. It produces a toxin. It's pretty nasty. But what they can do, so here is the genome is a circle, right? So it's a circular chromosome. They start almost like a clock at time 0. So these numbers here on the inner circle of this genome are minutes. And they start by allowing bacteria to conjugate. So they get them together and they let their pilus form, and then they kind of start sharing things, and they break that pilus at certain intervals, like one, maybe every minute, maybe every 10 minutes, whatever. And so then they can analyze what got transferred over to the new bacteria and say, okay, so in this particular time frame, if this gene was transferred early on, then that's going to be really close to the fertility factor. If it took 90 minutes before we could see evidence that that gene was transferred, then it's going to be a lot further away from the beginning of the transfer of that plasmid or that chromosome. So they've mapped operon. So there's the trip operon, there's a histidine operon, These are probably amino acid, other operons. So by purposely breaking that conjugation pilus at set time frames, researchers can map out on the chromosome where these genes are located. And I know you can't see the names on the E. coli O157H7, but I just wanted to show you how much they know about the genome based on kind of this method of mapping it with conjugation, which is kind of cool. All right, our last topic is those weirdies I said at the beginning. These are what we call transposons. And sometimes people refer to them as jumping genes. The woman who discovered these, her name was Barbara McClintock, and she worked with corn, various types of corn that had the different seed colors and things. But what she found was there's this gene, it's called the transposase gene, ASC ending, right? Transposase. And the transposase gene, once expressed, has the ability to make a copy of, so it turns into an enzyme called transposase. And that transposase enzyme has the ability to find the gene that codes for itself, copies it, and places it somewhere else in the chromosome. So it's like a self-replicating gene, which is weird to think that there's a gene that can control the location of itself with its protein product, its enzyme product. So let's take a look at what a transposon actually is. So we have the genes, right? The gene is this kind of box right here. It would be your sequence of A's, T's, C's, and G's that code for the mRNA, that code for the actual transposase enzyme. And then on either side, we have what's called an inverted repeat, sometimes called a palindrome. So this inverted repeat, if you take a look, we have A, C, T, T, A, C, T, G, A, T, and then upside down and backwards, A, T, C, A, G, T, A, G, T. So it's upside down and backwards. So that's what the inverted repeat is, and you have it on both sides. Okay. So let's say you have your chromosome with your transposon, this insertion sequence 1, IS1. What happens is it will get expressed, right? So it's going to get expressed and it's going to make its transposase enzyme. That's this big kind of tan blob. Once you have the transposase enzyme, it goes and it finds the insertion sequence. It finds the target sequence just somewhere else on the chromosome. It physically brings those two pieces together. It makes a copy of itself. It makes a copy of the target and inserts it right next to the target. So here's what we started with. Here's our target. And this is the product of transposase activity. Is it makes a copy of itself, it makes a copy of the target, and inserts it right next to the target. So you can imagine that this could cause problems because the targets are randomly selected and the transposase enzyme inserts two huge genes right smack dab in the middle of the target site, which could be a gene. So we could have a huge mutation or completely knocking out of a particular function of a gene. And so over here in the picture on the right, in this picture A, we could have one transposon. But then because of the nature of transposon, it bops itself over here. And now we have two genes for transposons. And now we're producing transposase enzyme, which will then copy and jump to other locations on the chromosome, right? So now it's like a self-replicating gene. And sometimes that can bop it over onto a plasmid. And now that we bop it onto a plasmid, the plasmid could be shared via horizontal gene transfer with conjugation, or maybe if the cell breaks open, the plasmid could be picked up by transformation that has this transposase gene in it, which will then self-replicate it in the new cell. So it's kind of crazy. We have transposons. This is not unique to bacteria. And a lot of human diseases are actually been known to be caused by transposons because they jump themselves right into the middle of a functional gene. We've lost that gene functionality and it ends in a disease, lots of cancers and stuff. This last piece down here is called a complex transposon. So a complex transposon can actually share other types of genes, not just the transposase. Right? So this guy down here. So what a complex transposon is, it's basically two of what we just talked about, an insertion sequence 1 and another one. And then right in the middle, in this case, they're putting a gene for kanamycin resistance. Kanamycin is a type of antibiotic. So not only is this going to be copying itself, but now it will copy the kanamycin and make replicates of that on the genome, which can then spread via horizontal gene transfer. So these weirdo transposons have the ability to when they get expressed, they make the transposase enzyme, which can then go and make copies of the gene that made the enzyme, self-replicating enzyme. Kind of wacky. All right, so that wraps up our chapter 7. I will see you next time. We have a little bit of chapter 8 that we're going to tag on to this genetics topic, a few pages of chapter 8. So we'll see you next time. Okay, bye.